Welcome to a new episode of Prime Movers, where we sit down with the founders, CEOs, and inventors who are transforming billions of lives. Joining us this week is LyGenesis co-founder and CEO Michael Hufford. LyGenesis is a clinical stage biotechnology company whose cell therapies use patients' lymph nodes as bioreactors to regrow functioning ectopic organs. Yes, you heard that right. The company is literally helping people grow an extra liver. Michael sat down with us to discuss the future of liver transplants and cellular therapies. Here's the full conversation. Well, Michael, thank you so much for joining us today. Welcome to the podcast.
It's a real pleasure to be here. Thanks for having me, Gavin.
No, of course. Well, kind of to kick things off, can you tell listeners a little bit about yourself and then dive into a bit about what LyGenesis is doing?
Yeah, so just by way of very brief background, I am a clinical psychologist by training, but I've spent the past 25 years now in drug development. And I've had a really eclectic career where I've had an opportunity to work on small molecules, peptides, proteins, medical devices. I've taken a number of things all the way through to FDA approval and commercialization, but I've kind of found my niche in working with small startups and kind of bridging that academic proof of concept to first clinical proof of concept, because that tends to be such a huge step up in value inflection. And just a lot of promising assets are lost in the chasm between sort of academic discovery and industry translation. So that's what I've ended up spending a lot of my career on. And then a little bit by way of LyGenesis. LyGenesis is a venture-backed cell therapy company. We focus on using the patient's lymph nodes as bioreactors to regrow functioning ectopic organs within their lymph nodes. And so it is a remarkable technology, one that I was very fortunate to partner with my chief science officer, Eric Lagasse, and chief medical officer, Paulo Fontes from the University of Pittsburgh, who had done all that basic scientific discovery, but really is enabling transformative regenerative medicine outcomes and generating these ectopic organs, and liver is our lead indication, which I'm sure we'll talk more about. But that was the stuff of science fiction just a few years ago. So it's tremendously rewarding and just a great deal of fun to work on technologies like that have such tremendous clinical potential and so much innovative science driving them. It just ends up being a tremendously rewarding area of work.
You also had the unique distinction of being the CEO of two different Prime Movers portfolio companies at the same time. It's usually, we like to back multi-time founders, but it's not usually at the exact same time. And maybe you can discuss your work with the morphoceuticals just a tad bit too.
Yeah, sure. To your point, I was very fortunate to meet Mike Levin and David Kaplan from Tufts University. Mike and David have really pioneered a radically new, really understanding of biology and focusing on the bioelectrics and the bioelectrome in particular. We have a lot of fascinating biological readouts these days, right? I mean, from transcriptomics and RNA-seq, there's lots of kind of read outputs to biology right now. What's remarkable about the work of morphoceuticals, that as you noted is now Prime Movers portfolio company, morphoceuticals is really enabling right access. And Mike's work has fundamentally helped us appreciate the role that bioelectrics play in really top-down governance of a lot of biological systems. So if you think of a lot of our genetic controls and genetic editing as bottoms up, trying to fix very specific genes that we hope will have specific biological outcomes, Mike's approach is really fundamentally different, is really fundamentally tops down, trying to understand the original biological drivers and the bioelectric control of things like organ generation and regeneration. And Mike's approach has been able to, for instance, regrow functioning limbs and regenerate limbs in animals that don't naturally regenerate. And that's all through this bioelectrics control and understanding the bioelectrome. So likewise with LyGenesis, that was sort of an early stage academic discovery. And so worked closely with Mike and David and their colleagues at Morphoceuticals and Prime Movers Lab, got the initial capital into that company, and Juvenescence had been really seminal in kind of identifying that work. Alex Pickett at Juvenescence had sort of come across Mike's work and appreciated the potential impact of Mike Levin's work. And so that's been a remarkable journey, and now they're off and running to the races as well.
Yeah. Circling back to LyGenesis, can you kind of just give us a little more, or why livers, and kind of give us an update of kind of where things stand? You're in clinical trials, and love to hear as much as you possibly can discuss about that, kind of where things stand at the moment.
Sure, absolutely. So LyGenesis was founded on the work of Eric Lagasse at the University of Pittsburgh. And what Eric discovered was that just as cancer probes its environment and often finds the lymph node a very hospitable reservoir to grow tumors, Eric discovered that you can place a variety of different cell and tissue types within the lymph node, and the lymph node will serve its biological and evolutionary function, which is, naturally it's a bioreactor for our T cells to help us fight infection. But if you put a lot of different tissue types in there, it will bioreact those cells and grow functioning ectopic tissues that can actually have a therapeutic effect. And so Eric is a world thought leader in the use of hepatocytes and how you can take that hepatocyte, which is the primary kind of functioning cell of the liver. There are many, of course, but that's one of the key primary ones. And it was his discovery that if you put hepatocytes into lymph node, over time, those hepatocytes would divide, they would form liver lobules and central veins, and remarkably, through a close connection to the native liver, would grow an ectopic organ that would support that diseased liver. And so when I first met Eric, he showed me the most remarkable data where he had rescued all of the animals, not just some of them, all of them, that would otherwise have a fatal liver disease by engrafting hepatocytes into their lymph nodes. And I remember asking him, saying, well, that's incredible data. He had just published it in Nature Biotechnology. And I asked him what I thought was the gotcha question. I said, well, but what about large animals? And he's like, oh, yeah, I can't get this nut to work. He's like, no, so not only did he have this remarkable data in mice, but he had also worked with colleagues at the Mayo Clinic, had replicated the data there, rescuing pigs that, again, have a 100% death rate from this genetically engineered liver disease, and was able to rescue them using this engraftment of hepatocytes. And as a drug developer, you don't often hear, I can't get this not to work. Now, that statement, I should know too, right? It's important to not just be good, but to be lucky in your career. And this is a case where a lot of evolutionary biology has set LyGenesis up for this finding, right? Ultimately, it was evolution that designed our lymphatic system to be this powerful bioreactor of the T cells. And of course, we've known since the myth of Prometheus, right? Since the Greeks, we've known that the liver will regenerate if given the right environment, right? So if you remember your Greek mythology, right? Prometheus stole fire from Zeus and gave it to man, was punished by having that eagle come and eat his liver out each day, but it would regenerate and grow back, right? And the core of that story is an understanding that's thousands of years old, that the liver will regenerate if given the right opportunity. And so one of the things that excited me about LyGenesis is how much natural biology it had going for it, right? You have the natural biology of the liver, where it will naturally regenerate if allowed to. You have the lymph node as this natural bioreactor that, again, exerting profound biological effects in a variety of different tissue types. You have the hepatocyte, which is kind of nature's stem cell. It is a natural stem cell that, through this close biochemical connection to the native liver, will help grow these ectopic organs. And so that's why our lead program is in the liver. And so we are a clinical stage company. If you're not familiar with us, so we're currently a phase 2A trial in patients with end-stage liver disease. We have now transplanted our first 4 patients in that first-in-human clinical trial. As a drug developer, it's both enormously gratifying, but the source of a lot of sleepless nights when you do first-in-human work. And so it's been a huge relief that all 4 patients have done extraordinarily well. In fact, I'm getting out in front of my skis a little bit because we're going to have a press release to this coming out soon, but I'm happy to share with you that we had an independent data safety monitoring board meeting just very recently. And they voted unanimously that the trial should continue and dose escalate as we had planned. So the first 4 patients got the low dose. The next 4 patients in the trial will get a medium dose. So they're going to get about 150 million hepatocytes engrafted into up to 3 lymph nodes. I should note we do that via endoscopic ultrasound. So it's also an outpatient procedure done under light sedation. That's key, both because clinically it allows patients that would not otherwise qualified for a transplant to be able to receive the therapy because it's low risk and minimally invasive, but also eventually from the economics of this, which we should probably talk about, that one of the things that LyGenesis has going for it is that this is also an extraordinarily cost-effective therapy. We can talk more about that. In terms of the trial, so the first 4 patients have done extraordinarily well. Safety and tolerability look excellent. We're now dose escalating to the next 4 patients. We've currently enrolled 1, in the process of enrolling more. Once those 4 are treated and we follow for 3 months, the Data Safety Monitoring Board will meet again. Hopefully we'll enable our final dose escalation to treat a final 4 patients. So this is a small N of 12, first in human trial. But insofar as we're trying to regenerate, ectopic organs in patients that are in desperate need of them. It is enormously gratifying and rewarding work that way.
Yeah, I can only imagine. Yeah, and let's dive into that, the economics a bit, or just the intricacies of it, because there are 10,000, 11,000 liver transplants in the U.S. every year. A lot of those are not successful. The body rejects it. The liver's too fatty. A lot of potential things, but you guys can have a unique way to solve it because it is the patient's own. It has grown in their body. So can you kind of just discuss how that really changes the economics of it, efficiency of the liver transplant process, and so much more?
Yeah, no, that's it. It's a great question and great point. Let me start by kind of making a kind of tip of the iceberg observation. So you're right, about 10,000 transplants, right? A lot of that was pioneered here at the University of Pittsburgh by Tom Starzl. And so there's a rich legacy of the treatment of liver disease coming out of the University of Pittsburgh here. That being said, that is tip of the iceberg, right? At least half of the patients that are on the transplant list, like for every one patient on the transplant list, there's at least one, if not more, that don't qualify for the list. So a lot of folks are aware of the tragedy that approximately 24 Americans die every day waiting for organ to become available on transplant lists that are not. So that itself is a tragedy. There is a hidden tragedy, though, that a huge number of patients don't even qualify for the list because they're so ill, because they have medical comorbidities where they simply won't survive the transplant procedure. There's this huge group of patients that need a transplant, will never get one because they're never even going to make the list. Behind them, you then have this epidemic of what's called MASH, this metabolic associated steatohepatitis, but basically kind of fatty liver disease, where you have this growing body of patients, many of whom are unaware that they have a liver disease until very late in its course. That also tends to a poor prognosis, and you end up treating them much later than you would like. And so among the benefits of our technology and the use, and to kind of bring it full circle to the use of endoscopic ultrasound, because we're not opening up these patients, we're going down through their mouth, right, with an endoscope. So this is an outpatient procedure. It takes about 10 minutes under light sedation. So the endoscope goes down through their mouth. It kind of transverses their gut. So if you imagine this is their gut wall, and the endoscope is going down, and you can image along the gut wall until we find a lymph node. And then we engraft our cell therapy into that lymph node, withdraw the needle, pull the endoscope out, and you're done in about 10 minutes. So that ends up being a procedure that from a Medicare perspective is billed at about a couple $1,000s, right? You compare that to the cost of a full liver transplant. Those acute hospital costs are just about $1 million, right? So you're talking about a procedure that is, many orders of magnitude less expensive to deliver the therapy. And then if you want to talk about a great just kind of startup metric, right? Today, one donated organ treats one patient. Using our approach, one donated organ could treat 75 or more patients. And that's because each donated liver is made up of billions and billions of hepatocytes. We're engrafting just a few 100 million hepatocytes, which over time will double and form these ectopic organs. But if you think of them as sort of the seeds that will eventually grow a meaningful, massive liver, hopefully, in these patients, the economics, the sort of supply-demand calculus is sort of fundamentally disrupted. Why are there only 10,000 transplants today? Well, at least one reason is there just aren't enough livers for everyone who needs them, right? Many of the livers that are donated today are not necessarily of great quality, so a lot of them cannot be matched, right? And so for us, being able to use donated but unmatched organs, and from a commercial standpoint, being able to eventually cryopreserve those and have an off-the-shelf solution, this is where you can really imagine, I wouldn't say the end of liver transplant wait lists. I would say for patients that need a full liver transplant, so like if you're an acute liver failure, our therapy is not going to have enough time to save you. So there's always going to be a role for liver transplants. But what we like is to have the use of the donated organ such that anyone who needs one will be able to access our therapy as either a bridge to a full transplant or hopefully as a curative therapy for their end-stage liver disease.
And you mentioned the Medicare cost of this. Can you kind of dive into that, just the impact on other stakeholders? I mean, you're partnering with hospitals who will be kind of developing the gene therapy there, how that works with, with their patients, and then just the impact this has on insurers, on the providers. I mean, I think you've done a good job kind of outlining the impact this has on the patient, but just all the other stakeholders in the ecosystem that are
sure.
in this.
It's a great question. For all the promise of cell therapies, and like you, I track, it's such an exciting time, right, between gene therapies and CRISPR and CAR T therapies. Fact is, many of those therapies are extraordinarily expensive. And there's no easy way to scale, especially for some of those therapies that are really per patient, unique to each patient. The issue of being able to scale those and make those cost effective is an enormous challenge. Because we're using these donated but unmatched organs, we have the ability, on a per patient basis, this therapy is going to cost on the order of thousands of dollars per patient, right? That's on the cost, right? So when you think of on a margin-based perspective, if you have a xenobiologic approach, the way a lot of folks are using CRISPR to try to humanize pig organs, incredibly sexy technology, very exciting technology, extraordinarily expensive, right? You have all the costs of genetically engineering the animal, harvesting the organ, and then the full transplant costs of the animal organ, right? So that is just fundamentally an extraordinarily expensive proposition with very little room really for any sort of margin to just make it a good business case. I think one of the things that's exciting about our therapy, we could have, potentially software-like margins, because then an off-the-shelf cryopreserved hepatocyte delivered in the way that we're delivering it means that we can deliver this therapy in a way that's extraordinarily cost-effective. And yet we'll both still have a margin important as to be a viable company and therapy, but also, you kind of fundamentally have the ability to treat so many more patients at an absolute cost that's very little. So for an insurer, we're going to be a fraction the cost of a liver transplant, and yet still have an enormous margin built in within even that cost. And so that's one of the things that I find appealing, that you have patients that are otherwise on an unfortunate path of eventual decompensation in terms of their liver disease, where they're going to start incurring lots of medical costs, right? And so it ends up being a very expensive end of life therapy. And so we have the potential to help insurers avoid all of those costs, and yet also come in at a fraction of what the cost of a full transplant is. And so we think and hope that we're kind of threading the needle in a way where you're delivering tremendous clinical value. You're able to treat patients that would not otherwise be treated, but you're also avoiding the very expensive end-of-life costs that are associated with slowly decompensating liver function.
Right. Some people listening may say, oh, it's only 10,000, 11,000 liver transplants in the U.S. It doesn't seem like, it's a huge market, but you can dive into the impact of like hepatitis B, which, affects 250 some million people around the world, and
exactly.
really just how broad the applications can be.
Yeah, no, that's a great question. And that's why I say like the liver transplant is such a powerful sort of human story, but the broader public health impact here, conservatively, we have hundreds of thousands of patients in the U.S. that would qualify for this therapy, right? So it's not going to be just folks that are on the transplant list. It's going to be folks that don't otherwise qualify for the transplant list. We can talk about, Hep B is, you have great vaccination rates of children, but very poor rates in adults. And although there are effective therapies, you end up with hundreds of millions of people around the world suffering from Hep B. You think about Hep C continues to be a problem. Alcoholic hepatitis is a huge problem, especially here in the U.S. And so, we've very intentionally tried to keep the sort of top of the funnel of patients as wide as possible. So we really let folks come in with a wide variety of potential liver diseases, because we know the unmet need is so great across all those various areas. And so the goal for our program is eventually, right, to have an FDA-approved biological therapy where you're going to be indicated for patients with end-stage liver disease, and trying to be as agnostic as to the pathophysiology as possible, because fundamentally, when you have a diseased organ that's trying to regenerate itself, that whole biological system is primed to grow these ectopic organs, because the diseased liver is emanating dozens of pro-growth signals. What we're doing is sort of hijacking the lymph node with our cell therapy to enable the liver to do what it's desperately trying to do, which is to regenerate it.
Yeah. And we've talked a lot about the liver today, but I'd love to kind of dive into the rest of the drug development pipeline you guys have going, because you have cell therapies potentially for develop like an ectopic thymus for aging and multiple, other potential indications for pancreas for type 1 diabetes, kidneys for end stage renal disease. Can you just discuss like all of these other potential applications?
Yeah, well, and a mistake I often make is I get so excited about our liver program, and it's very easy to sort of undersell some of the other pipeline work. At the end of the day, as a small company with limited resources, right, like you obviously have to place your bets. And so we have a clinical stage program, we're in patients, so that's enormously gratifying. Eric's discovery, though, that he's published widely on, shows that a variety of different tissue types thrive within the lymph node. So he's shown that he can rescue animals with diabetes by engrafting pancreatic islets into the lymph node where they glucose sense, insulin secrete, and can rescue those animals. And when you look at some of the money and interest from a drug development perspective right now in various pouches and ways to kind of protect islets that are being engrafted, including by some large companies now, I think the lymph node is an incredibly attractive target and place that you can engraft those islets where they can exert these therapeutic effects in a way that is very much aligned with the body's natural biology. So very excited about the pancreatic islet program. We also have a thymus program. So the thymus, for folks that aren't aware, plays a key role in aging, so that by the time you're middle age, your thymus has largely involuted and it doesn't exert a lot of biological effect, and in late middle age really becomes all but useless. It is a key way in which your body educates the T cells. And so one of the reasons that you end up with greater autoimmune disease and cancers later in life is that fundamentally your thymus is less and less effective as we get older. In Eric's lab and through some funding that we've received some non-dilutive funding from the Gates Foundation, in Eric's lab, he has shown that if you transplant an old animal with a young animal's thymus, it really shifts the entire transcriptomic profile of that older animal toward a more youthful phenotype. And so we're in the midst now of trying to document the beneficial immune effects of that. But its promise is potentially being able to reboot the thymus would have critically important potential implications for aging and immune function, and even things as routine as vaccine response all could potentially be enhanced through the use of an ectopic thymus. And then lastly, Eric's also published widely on the kidney, that it is possible to actually transform lymph node into an ectopic kidney that will actually produce urine. And so the potential there for end-stage renal disease, again, is incredibly exciting. There's some basic research to do in that area, but again, Eric's published widely in that. And so it's the challenge of being a small company. You would love to have adequate resources to kind of push forward on all four of those programs. We try to find research partners, funding partners to kind of move those forward, while clearly, the bet's been placed to a certain perspective on the liver as the most obvious kind of thrust of our work.
Yep. A lot of attention on work of Doge right now. So I'm always curious to get founders' opinions about their interactions with regulators and what things, they've learned working with the FDA in your case. I mean, are there any hurdles that you guys have had to overcome on the regulatory front that you think are, obviously so many are helpful, but anything else that's kind of maybe you almost want to suggest Elon to take a quick look at a few of these things because we've had a few founders actually engage with the administration on just some of these regulatory supersonic, for example. Blake Scholl was there this last week to discuss about lifting the overland supersonic regulations that exist. But are there anything like that you've encountered in your work?
I've had the opportunity to interact with the agency on a number of different programs, both in the biologics and new drug division. I would counsel that it's important with the FDA and the critical role they play. While there's always room for, efficiency enhancements, including at FDA, I'd also say you cannot throw the baby out with the bathwater, right? So we have largely, and I would agree with Scott Gottlieb, we have a largely functioning FDA system in terms of the review of the safety and efficacy of drugs. And they've done things like the Regenerative Medicine Advanced Therapy designation that's specifically designed to help accelerate truly transformative biological cures forward. And so there's a lot of good mechanisms for that. And we've had very good interactions. We've had very supportive interactions with the agency to date. And so, as I say, while there's always room for kind of improved efficiency, any attempt, I think, to fundamentally gut the FDA, I think would be a tragedy for American public health. There's, I could complain about, various FDA, interactions or, individual decisions I've had all day long. In the grand scheme, though, right, industry left unregulated would be an absolute disaster for this country. And I think largely you have a review group at the FDA that's working very well. And so it is my hope that, as I say, we don't throw the baby out with the bathwater there.
Yeah, no, understandable. Because we've had so many portfolio companies work through the breakthrough device program and others, and it's just fantastic that they've, are thinking about this and really trying to treat some of these breakthrough medical devices a little bit differently, but to actually allow lessons to be learned. So, I completely understand. And it's fantastic that you've had those experiences. I think, we kind of wrap up here. We love to end our podcast every month with a little founder questionnaire. This used to be our featured founder questionnaire that we would do when it was just the blog back in the day. But I'd love to kind of just run through these really quick. What's the most important thing to you in life? And maybe it goes back to the question of why are you at LyGenesis right now? Why this of all the things you could be tackling? Why this right now?
Fundamentally, I think I feel a tremendous responsibility to make the most of the opportunities. And I think when you have an opportunity like at Morphoceuticals and including at LyGenesis to enable science that could transform the lives of so many people moving forward, I think you feel an enormous responsibility to do that and do it well. So that's certainly something that I'm in touch with.
Yeah. What was the least expected challenge you've had to overcome as a CEO or startup founder?
I would just say that Murphy's Law, you sort of think that's just a pessimistic way of looking at things. But, when you're running a clinical trial that has as many moving parts as we have, we were taking donated organs that come available literally 24/7, 365, trying to process those, get them to these patients. Any person unavailable in that chain, the whole thing breaks down, so it certainly has been an exercise in kind of Murphy's Law. Like, if it can go wrong, it will. We're learning a lot of great lessons, and it's helping mature us and get us ready for a commercial therapy. But, if you just wouldn't believe the number of things that can go wrong when you try to get cells from a donor in Indiana to a cell processing facility in Boston to a patient in Houston. If it can go wrong, it will.
What's a breakthrough science prediction you have? I mean, it doesn't even need to be in your sector. It's something that between today and 2050 that you want to see happen.
Well, there's a lot I'd like to see happen. I would bring it back, and we already talked about Morphoceuticals, but I do think, if you think about, is there going to be a new chapter of medicine that is written in the coming decades? I think the only thing I've been exposed to outside of LyGenesis that I think qualifies that, where really fundamentally could alter our understanding of medicine and regenerative approaches to a variety of therapies, I think it would be the work that Mike Levin and David Kaplan are doing at Morphoceuticals.
That's great. What's on your reading list for 2025? People always love to see what founders are reading and what's inspiring them.
I tell you what, I recently discovered Byung-Chul Han, a Korean-born German philosopher. Byung-Chul Han writes these 70, 80-page books that are just blistering. His one, The Burnout Society, is an incredible diagnosis of a lot of what ails us in our current society, and also has important prescriptions for how to kind of reclaim your sanity. So if you've not come across Byung-Chul Han, I check out The Burnout Society. All of his books that I've read of his are outstanding, but that's really been one that'll kind of blister the paint on your mind's wall, you know what I mean? So it's highly recommended, Byung-Chul Han.
Okay, we'll have a link to that in the notes of this. So if anyone's interested, please check that out. Michael, thank you so much. I think that's everything. I really appreciate you taking the time to do this. And I know listeners have been absolutely fascinated by this. So thank you again. Really appreciate it.
Oh, it's a genuine pleasure, Gavin. Thanks so much.